Hi, everyone, and welcome to the webinar. We're so thrilled to have you today, to have you with us today. We want to share, we thought for September Blood Cancer Awareness Month, it would be highly appropriate to share all the innovation that's happening in multiple myeloma, lymphoma, leukemia, and MPNs. And so we brought several doctors together to do this for you in celebration of Blood Cancer Awareness Month. It's imperative that we share what we can about these diseases that we have to increase awareness and to advance research. So we're going to be talking a little bit about that today, a lot about that actually, as we also hear from these doctors who are amazing. Go ahead and advance, Marcel. Okay, we talked about this already. We'd like to welcome you to the HealthTree community. We began in multiple myeloma. We have expanded to these additional diseases, and many of the support services that we provide are available in these diseases. So we're great. It's so nice to have you, and we have over 700 people registered for this webinar, so it's fantastic. Thank you for joining. Okay, I'd like to start by just introducing HealthTree. We work really in three pillars, and I am Jenny Ahlstrom. I'm the CEO and founder of the HealthTree Foundation. We provide lifetime personalized support and education, meaningful patient-to-patient connections, and a powerful patient data portal. Why did we create HealthTree Foundation? Well, I'm a myeloma patient. I was diagnosed in 2010 and came from a tech background. My husband came from an entrepreneurship background, and we just thought things need to be changed because the current process of how research is performed, in my opinion, and many others' opinions, is just too slow. So how can patients contribute to their own cure? How can we provide tools and technology to the researchers to have them do their research faster on behalf of us so they can cure us faster? I was diagnosed back in 2010, like I mentioned. I did tandem stem cell transplants, a variety of combination therapies, and over almost 3 years ago, did a CAR T therapy. So I know some of you are very familiar with those protocols, and I've been very blessed to have my myeloma, for the large part, be in remission, so I can do this advocacy work and support patients. And it's been one of the most meaningful and rewarding and fun things I've ever done. One thing that we'll focus on a little bit later is the fact that we have technology. So it's very unusual to have the level of technology that we have built inside of an advocacy organization, but with over a very large team of software developers, we have provided over 14 different software tools that help empower you as a blood cancer patient to educate yourselves, to get connected with other patients that are like you, to share your experiences with those patients, and then to become active contributors to life-saving research. This is an example of how we use our education. We have many programs, as you can see. We have news websites, a university program, webinars, a podcast series, a specialist directory, sometimes live roundtables in myeloma, virtual roundtables in other cancers, and then live support with patient navigators. Our patient education events team is part of this event and has created many resources for you. This is Katie Braswell. She is our patient education director. And over her are Cindy, who is running the multiple myeloma webinars, Mary, who is doing the acute myeloid leukemia and MDS webinars as well, and educational content. Dalton, who is now doing our CLL and MF webinars and education content, and our operation managers, Marisol and Mary Lou. As our team has expanded into other diseases, we're adding additional employees to be able to support you. Now, on community, these are our patient-to-patient connection programs. So we have a HealthTree coach program where you can find a mentor. We have regional cure teams that we invite you to join if you'd like to participate in either advocacy, like handing out brochures, or helping people understand more about HealthTree Cure Hub, or doing local fundraisers. That's kind of our regional cure team. We have a REACH initiative where we're supporting, like for example, Black Myeloma Health, or a Hispanic program with content in Spanish. HealthTree Connect, which is our own social media platform, and then a HealthTree Moves app that will be used to help do community type get-togethers for fitness. You'll be hearing more about this, and I'll be doing a demo for you today about HealthTree Cure Hub, but this allows you the ability to contribute to academic research in significant ways. And we'll talk about why this has never been done before and why we're the first to do it. There are other benefits that you can see here about participating in HealthTree Cure Hub, but I want to spend more time on that after the doctors conclude speaking, so I hope you'll stick around for that. We'd like to thank our Blood Cancer Awareness Month sponsor for this series, GSK, and our event agenda is, we'll first start with Dr. Josh Richter, who will share multiple myeloma. Dr. Tremblay will then share leukemia update. Dr. Habermann will then share a lymphoma presentation, and Dr. Mascarenhas will share on for MPNs. And then I will show you how you can play a role in advancing blood cancer research through HealthTree Cure Hub. And with that, we will get started with our doctor update.
I'm excited to introduce our first guest speaker for today, Dr. Joshua Richter, who will speak to us about innovation in multiple myeloma. In interviewing Dr. Richter, I learned he has a knack for explaining complex things in a way that is easy to understand, but also brings with him a wealth of knowledge that he has accumulated in his years of teaching, treating, and researching plasma cell dyscrasias. He has published in journals such as the New England Journal of Medicine, Blood, and the Journal of Clinical Oncology. He's regularly asked to speak on his research, which is focused on novel therapies, including antibody therapy and immunotherapy, for a precision medicine approach. We're so lucky to have him share his time with us as we round out Blood Cancer Awareness Month with this webinar. With that, I'll turn the time over to Dr. Richter.
My name is Dr. Joshua Richter. I'm an associate professor of medicine at the Tisch Cancer Institute, Icahn School of Medicine at Mount Sinai, and the director of myeloma at the Blavatnik Family Chelsea Medical Center at Mount Sinai. And I'm really excited to talk about some of the amazing innovations we've had across the last number of years in myeloma and what the future looks like. So in general, I think about the three great epochs of treatment in myeloma. The first one started when we first started treating cancer in general, and the first epoch was classical chemotherapy. And in the old days, we didn't really have a lot of tools in our tool belt, and we didn't have some of the amazing advocacy groups that we have nowadays, like HealthTree, really getting together researchers from around the country and around the world to really help fight and cure myeloma. And back then, all we had were the classical chemo drugs like melphalan or Alkeran, as it was given as pills. And there's an older regimen called VAD. VAD was infusion of vincristine, adriamycin, and dexamethasone. Pretty rough stuff. And then we had a couple other chemotherapy drugs like cyclophosphamide. And then in the 80s, we really started to do high-dose chemotherapy with stem cell transplant. But really, all chemotherapy is giving measured doses of poison. These drugs are not specifically targeted necessarily for cancer. What they do is they kill cells that divide rapidly, because in general, cancer cells divide rapidly without listening to the body's cues for when to kill themselves. And the side effects kind of make sense, because what cells divide the most rapidly in us? Well, the hair on the top of our head and the lining of our GI tract. That's why classical chemotherapy makes us lose our hair and have GI symptoms like nausea and vomiting. Well, that was the first epoch. And then right around the 90s and 2000s, we really started getting in to the age of the second epoch, novel therapies. And even though these drugs are more than 20 years old, we still call them novel. And this really started off with some of the amazing work that was being done in places like the University of Arkansas with Dr. Barlogi, with drugs like thalidomide and analogs of thalidomide. And if you remember thalidomide that was given to pregnant women here in the US and many places in Europe in the 50s and 60s to help with sleep and morning sickness. And one of the reasons the drug was given is that classical sedatives that help actually suppress the respiratory rate. So you have to worry about that when you have a baby in you. But thalidomide, no matter how much you take, it doesn't affect the respiratory rate. So they thought this would be a good drug for those women. Well, it turns out that that drug does many things. And one of the things it does is inhibit vascular endothelial growth factor, or VEGF. And that's basically helps little babies grow new arm buds through the growing of new blood vessels. And unfortunately, some of those children were born with birth defects. Well, even though it kind of sounds weird, the formation of new blood vessels is important for blood cancers, not just for solid tumors, because they can spread inside the bone marrow through these new novel blood pathways. Well, thalidomide does other things. It affects our immune cells and actually doesn't just ramp down the bad stuff, it actually helps to ramp up the good stuff. And we started calling these drugs immune modulators, or IMIDs, because they modulated the immune system. They helped our own body fight the cancer. And then we started getting newer versions of this, like Revlimid or lenalidomide and Pomalyst or pomalidomide. And these drugs are amazing at not just killing the bad stuff, scorched earth like the first epoch with classical chemo, but they suppress some, it targeted some of the cancer cell, but also helped to boost a little bit of our own immune system to fight it. And that was kind of giving us kind of sneak preview about the third epoch. But still in the second one, we had some of our novel agents, and then we got the proteasome inhibitors. And proteasome inhibitors are really amazing drugs because basically they target the garbage disposal system of myeloma cells, and all cells actually. Well, all cells make garbage. Myeloma cells make the most garbage. That's that M spike, that N protein, or the free light chain. That's the garbage. And if you stuff a shirt in the garbage disposal, the cell fills up with the bad protein and explodes. And now we have 3 FDA-approved proteasome inhibitors: Velcade or bortezomib, Kyprolis or carfilzomib, and ixazomib or Ninlaro. And all of these drugs kind of work the same. And then around the year 2015, we got one of the most amazing drugs of the novel age, which was Darzalex or daratumumab. This is a monoclonal antibody. Now, our body makes antibodies all the time to fight viruses, bacteria. Well, what daratumumab is, it's an antibody that's targeted against your myeloma. All cells in the body have markers that stick up on them, and we call them CDs, or clusters of differentiation, because they help us differentiate one cell from another. And for example, myeloma cells have CD38, and they have kappa, and they have CD269, and it turns out CD38 is a great target. And that daratumumab drug targets CD38, so it locks onto the cancer cell, helps to kill it, and actually does help to modulate our immune system as well, and grabs along some other cells and say, hey, here's the bad guy, let's go fight the bad guy. And this drug really changed the way that we fight myeloma. And one of the reasons why the novel agents change things so much is our ability to combine them. We know about myeloma that myeloma is not one disease even within an individual. In fact, we know that the disease is made of subclones. And at the time of diagnosis, most patients have between 4 and 6 subclones. Well, we know that the deeper remission you get correlates to durability. Well, how do we get a deeper remission? Kill more subclones. How do you kill more subclones? Multiple drugs with multiple mechanisms of action. So drug 1 kills clone A, and drug 2 kills clone B. And that third clone, you need the synergy of those 2 drugs to kill it. Well, in the days of classical chemo, that's a big ask. When you start adding 2, 3, and 4 chemotherapy drugs, that can be very, very toxic. But one of the things about the novel agents is we learned we can combine them. We can combine drugs like daratumumab and Velcade and Revlimid and dexamethasone to really now the standard of care for many of our newly diagnosed patients is giving 4 drugs at the same time. 4 drugs allows us to kill more subclones, get a deeper remission, and therefore a more durable remission. And if we think about how much this means, let me take you back to the 1970s and 1980s. In the 1970s and 1980s, the entire journey for a myeloma patient, from diagnosis to death, all comers, doesn't matter if you were old or young, fit or frail, rich or poor, was 2 years, overall survival. We recently had some data presented from the IMRAJ trial. This is giving a 4-drug combination, isatuximab, another CD38 targeting monoclonal antibody, with Velcade, Revlimid, and dexamethasone, to our older transplant ineligible patients. And the median progression-free survival is around 90 months. That means that there are many patients who are going to be diagnosed now with myeloma who are too old or too frail to get a stem cell transplant, but will never relapse in their lifetime from myeloma. And although that's not a cure, that's a really important step towards cure. And it's really getting many of our patients to a functional cure, that if we're able to take your disease and put it in remission and you never relapse, that is a version of a cure. But that's still just the second epoch, because in the last few years, we have reached the third epoch, the most amazing responses that we've ever seen in myeloma with the T-cell redirection age. And this is where we really are saying, well, the best cancer fighter in the world is our own immune system. In fact, everyone on the planet makes cancer cells 24 hours a day. The best way of fighting cancer is our body's mechanisms are set up to say, no, you're cancer, you go commit suicide. Well, we started off with chemotherapy, killed the good and the bad. Then we went on to novel agents, which killed some bad and helped the good. But what if we took all of our efforts to just ramping up the good? And that's what the third epoch is all about. Using therapies like bispecific antibodies and CAR Ts to take our immune system and put it into hyperdrive. The best way to think about it is think about our immune system like the armed forces. You have the Army, the Navy, the Marines, the Air Force. What if we took a bunch of those, first-day recruits out and we sent them for special forces training, equipped them with the best technology, and then sent them out to fight? That's what we're doing. And the two technologies, one are bispecific antibodies, the other is CAR T. Bispecific antibodies are just like daratumumab and isatuximab. They're antibodies, but they have two arms. One arm grabs onto the cancer cell, the other grabs onto our own immune cell and activates it to attack the cancer. I think about this the way there's this old movie called Mad Max. And in Mad Max, with Tina Turner, who's amazing, they had this saying about Thunderdome, 2 men enter, 1 man leaves. That's what a bispecific antibody does. The cancer cell and the immune cell enter Thunderdome, and only the immune cell leaves. These drugs are unbelievable, and they're off the shelf. They can be given anywhere at any time, because myeloma doesn't always wait around for the right time and the right place. And if your disease is out of control today, we can give a drug like this today. Amazing. And some of the best therapy we've ever had is CAR T, chimeric antigen receptor T cells. These drugs go by the principle of, well, right now, your immune system, your T cells are fighting myeloma, but they're not enough, and they're not fighting hard enough. So what if we collect them the same way many patients get their stem cells collected? But we switch the dial on the machine, and instead of collecting stem cells, we collect your T cells, and then we send them out for special forces training, and we equip them with the greatest technology and greatest weaponry, and we make them target your myeloma. So one of the most important targets that we've identified in the last number of years is BCMA, or B-cell maturation antigen. It's another one of those CDs, CD269. And for 2 of the 3 FDA-approved bispecifics and 2 of the FDA-approved CAR Ts, that's the target they use. And that target is almost exclusively on the bad cells. It's a little bit on some other immune cells, but it's really mostly on the cancer cells. So now we collect your T cells, we engineer them, but we can't just infuse them into your body. There'll be a voice amongst the crowd because we all have a T cell repertoire. We have T cells in our body fighting bacteria, viruses, cancer. We want those T cells to be the dominant force. So we give low doses of chemotherapy to create immunologic space, and then we reinfuse those CAR T cells and they attack the cancer. It's a living drug. And the best part about it is that the kind of mantra for the last decade and a half of myeloma has been treat to progression or intolerability, which means people are on constant treatment for years. That's a huge toll. And one of the amazing things about CAR Ts is that we call it a one and done. You get the CAR T and then you don't get constant therapy. The drug or the therapy that lives in you continues to fight the myeloma. And the hope is that either it gets the disease so far down that it stays down really long or forever, fingers crossed, or it develops a kind of long-term memory response where it kind of helps your immune system say, hey, if you see this guy again, you've got to kill him immediately. And these therapies are amazing. And the future of myeloma right now is taking this third epoch, the T cell redirection, to the next height. And how are we doing this? Well, one of the things we know is that combining therapies in myeloma is the way to achieve those deep remissions. We knew that even back in the second epoch, where combining multiple agents was the way to kill multiple subclones. Well, what if we update that model and still combine, but combine with our newer optimized therapies? So now we have a couple of big targets we're going after. CD38 is one of them. BCMA is another. There's another one called GPRC5D. It's another marker that's on all myeloma cells. We even have a newer one called FCRH5. And we're starting to combine them in a number of really novel ways. One is by taking some of those bispecific antibodies and giving them along with drugs from that second epoch, combining a bispecific antibody with daratumumab, or combining a bispecific antibody with an IMiD drug like Revlimid or Pomalyst. We actually have a study called Redirect, where we take two bispecifics and give them together. And one of the things is we're seeing patients that have extramedullary disease, that's myeloma that lives outside of the bone marrow, that can be particularly difficult to treat. But when we give two bispecifics at the same time, it seems to melt away. We're having unprecedented responses with this approach. For CAR Ts, well, we're coming up with a couple of novel approaches to optimizing CAR Ts. One is right now they're all autologous, meaning we collect your T cells and engineer them to put them back in you. Well, that engineering can take time. So we have a couple of ones that are approved, but they can take 4 to 8 weeks sometimes to make. Myeloma doesn't always want to wait 4 to 8 weeks. So we have fast CARs, CARs that we can manufacture in less time, maybe a week or two. We have allo CARs, CARs from a donor that can be off the shelf and ready to go. Now we have dual-headed CARs, and we have a couple of really fancy dual-headed CARs. We have one dual-headed CAR that gets that BCMA and the GPRC5D. Those are two of our most potent targets to date. We have one really clever one that combines BCMA and CD19. CD19 is not normally expressed on myeloma cells. It's really common in lymphomas and leukemias, and there are several CAR Ts and bispecifics approved that target CD19 in lymphoma and leukemia. But there's some theories about a myeloma stem cell that expresses CD19. So the concept behind this CAR is you kill the main stuff and you kill the stem cell. Like you have a tree in your front yard that you want to chop down. You chop it down, but you also poison the roots. So one of these technologies may emerge as a pathway to either unbelievably long remissions or even cure. Because in myeloma, when you start off with a new therapy, we always have to start off at the end, because right now, our upfront treatments have about 100% response. And as I mentioned, with regimens like these quadruplets and IMRAs, 90 months without a transplant, well, we can't take a brand new therapy and just replace that, even though it's exciting. We test it, and then we say, all right, it works. Well, is it better than what we do in later line? Okay. Is it better than what we do in early line? Okay. Is it better than what we do in frontline? And right now, there's some really amazing studies like the CARTITUDE-6 study, because right now, the standard has been for transplant-eligible patients to get induction therapy followed by a stem cell transplant. Well, is CAR T better? We don't want to guess. We want to know. So it's a randomized study. Patients get induction therapy, and they get randomized to a stem cell transplant or a CAR T. So we're really on the precipice of achieving unbelievably durable remissions. And what we're going to start seeing over the next few years is a leap, not only in therapeutics, but in diagnostics. And one of the things that we've seen in the world of other malignancies like Hodgkin's disease is that the response rates and the cure rates are so high, a lot of efforts are now to back off. What is the minimal amount of treatment to cure? Well, we know that not all myeloma patients are the same. So do we need to get everyone to MRD negativity? We know that people who get MRD negativity, that means with our best technology, we can't detect any myeloma. They do the best, but that's not the whole story. And there's some amazing work coming out of other parts of Europe from a doctor named Bruno Paiva, who's noticed that some patients revert to what's called an MGUS-like state, meaning we get the myeloma down. It never goes to zero, but what's left doesn't do anything. It just sits there. So does everyone need therapy forever? No. Again, everything comes down to Goldilocks. We don't want the porridge too cold. We don't want you to relapse. We don't want it too hot, meaning we're giving you constant treatment for years, even if you don't need it. So the goal over the next 3 to 5 years is to make the porridge just right for everyone. We almost have this joke in lymphoma, they have hundreds of types of lymphoma and one treatment. Right? Everyone gets R-CHOP, even if you have diffuse large B or activated B cell or Burkitt-like, they have all this. In myeloma, we have 100 treatments, one disease, multiple myeloma. But what we're struggling towards over the next period of time is identifying what makes your myeloma different from someone else's and really giving a precision medicine approach that goes from soup to nuts, where you'll come in for a diagnosis. We'll send next level genetic sequencing, proteomics, immunologic sequencing. And instead of just giving a regimen because it has high response rates, we will begin to custom tailor this for you for either unbelievably long remission or cure. And when I started doing this, I would sit down with patients and say, unfortunately, this disease is incurable. And what I say now is we are currently curing a few patients by accident. We don't know at the get-go that we are going to cure them, but we end up doing that. And over the next period of time, we're going to start to do this more and more commonly, more and more intentionally. And the goal is we don't stop until we have 100% cure for 100% of patients, 100% of the time. So I think the future is incredibly bright. And thanks to amazing advocacy groups that are there and all working together on the same side between advocacy groups like HealthTree, patients, caregivers, industry, academics, community, we're all going to get the same goal. And with that, I thank you very much for listening.
Thank you so much, Dr. Richter. We have come a long way since the introduction of thalidomide in myeloma treatment, and with new targets, new combinations, and new therapies being developed, it sounds like there's a lot of exciting work being done to get us closer to a cure. Next, I'd like to introduce our Innovation in Leukemia speaker, Dr. Douglas Tremblay from the Tisch Cancer Institute at Mount Sinai in New York. Dr. Tremblay is a warm and welcoming physician who is known by his patients as someone who will truly take the time to listen to their concerns and help them come up with solutions. He easily explains complex ideas in ways that are simple to understand. With over 100 publications, Dr. Tremblay is well known and passionate about advancing leukemia research and doing it in ways that puts the patient and their quality of life first. We're so excited to have him talk to us today about the exciting things going on in leukemia research. With that, I'll turn the time over to him.
So my name is Douglas Tremblay from Mount Sinai, which is in New York City. And I'm going to be speaking about new innovations in the field of leukemia. So this is a really exciting time of leukemia, but in leukemia treatment development, but it's also important to reflect on where we've been, which is that the standard of care of leukemia hasn't really changed a lot for many patients. And despite the introduction of many new and excited targeted therapies, as well as the addition of another drug to non-intensively treated patients, there's still a lot that needs to be done to improve the really bad outcomes that are experienced by many patients with leukemia. And so I'm really excited to speak today on a few different areas of research that I think are very innovative and really have the potential to change the game for patients who suffer from AML in particular. So the first one I'm going to speak about is a whole new class of drugs called menin inhibitors. And it's really important to identify and really say from the outset that finding a new pathway and exploring that pathway is really exciting because it opens up not only the treatment for one drug, but often many different drugs that can really serve and help patients get into remission, live longer, go to transplant, and ultimately improve their lives. So it's important to say that AML, while it's defined as one disease is actually many different diseases. And some of those diseases have certain vulnerabilities with them where it makes it possible for us to give medications to really target those vulnerabilities. One of those vulnerabilities is a certain type of fusion called KMT2A. And this is a type of leukemia that is associated with prior chemotherapy treatment, is an aggressive type of leukemia, and has a very specific way that this sort of leukemia causes, or this sort of translocation causes leukemia. And that goes through a bunch of different expression pathways in the cell, like HOXA and MEIS1, that really drive this leukemia forward. And this sort of interaction between KMT2A and this molecule menin is really necessary for this sort of leukemia to develop. And so there's been a really great development of these new drugs called menin inhibitors, which really disrupt that binding of those two interactions and break up the kind of pathways that lead to AML to develop in those patients. And although KMT2A rearrangements are pretty rare in AML, about 10%, there's more commonly mutated types of AML like NPM1, which also have this sort of pathway that can be exploited and targeted with menin inhibitors as well. And we're learning that there's even other mutations that this type of interaction may be exploited as well. So there's now 2 menin inhibitors which are in later stage development. And these have been shown to have a response rate of around 50%, which, although it doesn't sound that high, is pretty monumental in patients who have relapsed refractory AML, and does allow many of those patients to move on to things that are more curative, like allogeneic stem cell transplant. There's also, in addition to these 2 menin inhibitors, revumenib and ziftomenib, there's 4 additional menin inhibitors that are earlier in development but have even more unique possibilities to try to target this pathway and really help people who have KMT2 rearranged or NPM1 mutated AML as well as other related mutations that cause MEIS1 expression or HOXA expression as well. But also really exciting is that this menin inhibitors is being combined with other drugs too in both the relapse setting as well as newly diagnosed patients, which can really, I think, enhance the ability of these drugs to work. And by putting all of these different tools together, it's possible to induce much higher response rates and eventually really take a crack at this really challenging disease to treat. So menin inhibitors is a new class of drugs which will hopefully be available soon, and look for this pathway to start being exploited more and more, and this vulnerability will be exploited more and more with new drugs that can really, really target this effectively. The next innovation I'll speak about is one that has been unfortunately pretty disappointing in the past, which is immunotherapy and CAR T cells. In other types of leukemia like ALL, as well as in other B-cell lymphomas, and there's CAR T cells have really redefined survival expectations in these diseases. But AML presents a lot of unique challenges for CAR T cell development, and specifically that it's really hard to find some characteristic of AML cells that makes it different than normal healthy bone marrow cells. So if you don't have something to differentiate these two, it makes it really challenging to give a drug that's not going to just be extremely toxic to the bone marrow. But there's new targets that are being explored that can really try to overcome this lack of specificity. And some of those new targets are looking quite promising and are further being developed. And there's also new approaches to the CAR cells and using different types of cells like CAR NK cells and fine-tuning CAR T cells to try to improve the sensitivity to the actual leukemia cells while sparing the normal cells. So this is much more early in its development as compared to menin inhibitors, but it's something that I think is really exciting to see, and hopefully in the future this will enter the treatment armamentarium as well. Another aspect I'll mention too is transplant strategies. Even though transplant can be a curative treatment for patients with leukemia, there's still a major limitation in that many patients will relapse, or there's issues like graft versus host disease. And so there's now promising avenues that involve engineering these sort of donor cells for the transplant so that they target any sort of leftover leukemia cells as well as any leftover stem cells from the patient and then spares any of the donor cells. And the idea is to really make it so that all of the patient cells and the disease cells are all erased, and that the donor cells can work much better at reconstituting the bone marrow and also fighting any residual leukemia. And there's a trial that is really exciting to see with a platform called T-Scan that results are very early, but are suggesting that this is really safe and efficacious ability to detect or eliminate any detectable residual leukemia cells, which should reduce the risk of relapse happening. So transplant is still going to be a part of our life for a long time in AML, I suspect, and it's important that the way that transplant occurs is being continually innovated on to try to make it more effective. The last big update I'll mention isn't so much a new strategy or anything, but is really, I think, a really monumental effort by many people in the leukemia community to come together to design what we call umbrella trials, precision umbrella trials for AML and MDS through a platform through the cooperative groups called myeloma. And an umbrella trial is a type of clinical trial that evaluates multiple drugs in patients with the same disease, but they may have different biomarkers. And this is a really innovative platform that is just starting to get going now, where patients will be able to be enrolled, and based on their exact characteristics will be matched with specific clinical trials which are targeted for that disease. And these different earlier phase 2 trials can then provide more information that then can be expanded into larger trials and really enhance and flesh out the treatment landscape of AML and MDS. So this has been a monumental feat that the NCI has really undertaken to try to develop this MyeloMatch platform so that there can be not just one new clinical trial, but dozens of clinical trials in different settings, either up front or at consolidation or MRD positivity or transplant, that we can really use that to efficiently enroll to these trials and really accelerate the therapeutic development for AML and MDS. So I want to highlight this because I think it's been a massive undertaking that's been happening for a long time. And it's now just coming to fruition where this platform is being open and these trials are starting to open. And it's really exciting to see how this will really step on the gas, so to say, in treatment development. So these are 4 areas, I think, of innovation that are happening in the leukemia space, some which are more sooner to come out and be available to patients, like menin inhibitors, some that are later on, like CAR T cells. But all of these are very interesting, innovative, and exciting prospects that make me really excited for the future for AML treatment, and really excited to be a part of many of these efforts and to be see how this field really develops.
Thank you, Dr. Tremblay, for that incredible presentation. We will be excitedly following the innovations in menin inhibitors, CAR T, transplant strategy, and precision medicine. What an exciting time in leukemia research. Our next speaker for today's event is Dr. Thomas Habermann. Dr. Habermann is a hematologist-oncologist at the Mayo Clinic in Rochester, Minnesota, specializing in the care of individuals with different types of lymphoma. Dr. Habermann is an incredibly accomplished and well-respected physician. He has been a key part of a lot of exciting innovation in the lymphoma space over the years. With over 400 authored papers and 7 books, he's a powerful voice in the hematology world. We are so grateful to have him here to talk about innovation in lymphoma. With that, we'll turn the presentation over to him.
Thank you. It's a privilege to be here and discuss innovation in lymphoma 2024. These are my disclosures. Innovations in 2024 come under 5 different areas in my judgment. The classification, what type of lymphoma, the biology, type, trying to find the genes and the immune cells associated with lymphoma, the treatment changing paradigms, toxicity, trying to improve in managing side effects, and the epidemiology, to study the distributions, causes, and control of disease in the population. Please listen and follow the big picture in this presentation. Follow the red colored words. Don't get hung up and lost in the acronyms, that is, the abbreviations formed from the initial letters of other words and pronounced as a word. And what's confusing to all of us are drug names and gene names. Lymphoma is complicated. There are so many subtypes. The change in pace of research is rapid. Lymphomas that are not curable are now becoming chronic illnesses. The biology is complex, the collaborations must be integrated at all levels, and the funding is multifaceted. How did these innovations happen? Patient participation has been really very, very critical. Clinical trials, consent to provide tissue for clinical research, follow-up surveys. There must be correlation with biology and clinical information, and a very large number of patients with clinical and specimen data are absolutely essential. Number one, classifications. There are now two classifications in 2022, the ICC classification and the WHO or World Health Organization classification. In the WHO 2022 classification, I count at least 118 different types of lymphomas. This was a study we did of almost 600,000 patients in the National Cancer Database in the United States, and the prevalence of the different lymphomas was as follows. Diffuse large B-cell lymphoma and its subtypes represented a third, and follicular lymphoma represented 17%, and so we'll discuss these issues. What about the biology? This was a study we were involved in the molecular subtypes of diffuse large B-cell lymphoma. 304 newly diagnosed patients were evaluated with genomic studies. There were 158 genetic driver alterations. There were 6 genetically distinct subtypes identified. And this is what the map looked like. Where have we gone from there? To advance our understanding, we developed a concept of event-free survival at 24 months, or EFS 24, and we've continued in the journey of multi-omic sequencing, including whole exome sequencing, that is sequencing of all the protein coding regions in a genome, and RNA-seq, which is transcriptomic approach where the total complement of RNAs is isolated. Achieving EFS 24 is landmark observation in aggressive lymphoma. In diffuse large B-cell lymphoma, patients who achieve an EFS 24, which means no relapse, no retreatment, and alive at 24 months, have a survival that's similar to the general population in the 5 years after achieving an EFS 24. We studied it with scans, and that was called PFS 24. Well, what does this look like? On the left is the overall survival from diagnosis. The red line is the age and sex matched population. The black line is diffuse large cell lymphoma patients, and you can see they don't do as well. But on the right, if you then start looking at overall survival after achieving EFS 24, you can see that the curves superimpose on each other. The next area we got involved in was a transcriptomic classification of diffuse large B-cell lymphoma. The goal of this study was to identify high-risk patients in diffuse large B-cell lymphoma. How did this get accomplished? We partnered with industry. A large number of patients were included, 3,192. Recall that I just said in the study in 2018, we had 300 and some patients. We used patient samples from a large number of clinical trials and cohorts. What was learned? A new cluster was demonstrated. Now we would suggest there are 7 clusters, and drugs such as lenalidomide, a drug that's been around, can overcome low tumor infiltration by a new subgroup by inducing T-cell trafficking into tumor cells and to upgrade MHC1 and MHC2 on tumor cells, so outside of the inside of the cell. And we demonstrated a regulator of MYC biology. MYC is very important in this disease. You can see in the curves on the bottom in the red line the inferior outcomes of these patients. We then went on and to study things further, because these genetic and molecular classifications that I just presented were not designed to define early events. So this study incorporated multi-omics, whole exome sequencing, and RNA-seq, and clinical data from 444 newly diagnosed large B-cell lymphoma patients. The inclusion of ARID1A mutations resulted in the identification of 45% of cases with an early clinical failure, and we validated it. This was the first study to identify a signature that identifies patients for early clinical failure. We included the concept of the EFS24, and you can see in the red line the poor outcomes of these patients versus low-risk patients at the top. And you can see there's a difference in 95% survival at 4 years in the top versus around 25% in the bottom. Well, what about follicular lymphoma? This was the largest study published at the time of looking at the patients treated in the rituximab era, and we reported that essentially 80% of patients were alive at 10 years. What have we been looking at further now? Now we're actually moving studies, RNA-seq studies, to isolated B cells from follicular lymphoma tumor patients. And we've been looking at, in the node, different traffic and different understanding of the geography of different T cell subsets and where they play out, and then the genomics on the right. What did we learn from this particular study? We identified 3 transcriptional states: inflamed, proliferative, and chromatin modifying. So why is this a big deal? Well, each of these 3 states was associated with a combination of mutations. The hope a long time ago was that we just would have certain mutations. Also, follicular helper T cell interactions, and then the microenvironment with activated T cells help identify 3 different subsets, which will be under further study in the future. Well, what about treatment? This is the 75 years of lymphoma treatment, starting in 1949. The second drug, methotrexate, in 1953, the year I was born. Then along came rituximab in 1997. The first era was the chemotherapy era. Rituximab started the targeted therapy era. And now we are in the immune therapy areas. And you can see in the blue drugs in the final right side that there are 12 drugs there that have met regulatory approval and under further evaluation and further study and further treatment profiles. We know that we can cure 65 to 75% of patients with diffuse large B-cell lymphoma. We can do that with standard regimens in orange that are difficult to see here, but R-CHOP chemotherapy, R-EPOC, Pola-R-CHP. But then where all of the efforts are now focusing far more is in the relapse refractory setting. We had transplant, we now have CAR T cells, and then underneath this you can see that we have in orange different drug regimens, and in blue different drug regimens. And these novel agents are available, but in what sequence and how are we going to use them? What's an example of a very recent change in the last 3 years especially is that in relapse refractory diffuse large B-cell lymphoma, if patients are fit for high-dose therapy and they are late relapse, then we still proceed to the auto stem cell transplant. In patients who are relapse at less than 1 year, then in those patients we will move to CAR T cell therapy. For those who are unfit, we then have the multiple other drug regimens that we potentially have available that I briefly showed you. This is what's happened in the CAR T cell era. These are the 3 Food and Drug Administration approved regimens or drugs, and the 2-year progression-free survival is 30 to 41%, so remarkably good outcomes. And now there are all kinds of different new CARs that are being developed and new sophistication because of this understanding. But along with this era, the bispecific antibodies, these other cells had to be manufactured and using patient cells, but bispecific antibodies are off the shelf. So a combined antibody of anti-CD20 and anti-CD3 and different immunoglobulin fragment formats have been developed and are under evaluation in studies and future studies. Mosunetuzumab is an example of a bispecific antibody. You can see this blue antibody here in gold. This attaches not only to the T cell, but attaches to the tumor cell And there is an immune synapse formation and cell death, and the complete remission rate in the initial trials are in the 19% range. Glofitamab is another drug that's used in combination with obinutuzumab, and it, in looking at the complete remission rate in all doses in all patient studies was 39%, and this is under continued evaluation. A second to the last area that's exciting is patient monitoring and toxicity. We put together an international collaboration of 40 patient advocates, clinical investigators, regulators, pharmacists, biostatisticians from multiple continents. This essentially represented the modern world worldwide, and produced a call to action paper with tangible targets and timelines for improvement. We want to improve patient experience data and how we get it, so Apple Watches and other approaches. We want to improve tolerability and survivorship. And we have 2 publications out, and we're now planning a series reflecting each of the 6 subtopic sections. Lastly, what about epidemiology? We developed the LEO Consortium, which is an integrated long-term follow-up cohort, 8 academic centers that are very interested in lymphoma. From 2015 to 2020, 7,735 patients were prospectively enrolled, and we continue to enroll. But most importantly, we're now really improving our accrual in underrepresented minorities, including Black African Americans, Hispanics. We've always accrued adequately in women, but we're really looking at any differences we can look at there. And we collect multiple sets of data, including serum, plasma, and genomic data from patients. And so with that, I think these are the innovations and directions that we're going in these fields. And I'd like to thank you for your attendance and your attention. And these are colleagues who I especially want to recognize. Thank you.
I'd like to thank Dr. Habermann for sharing his presentation about how much lymphoma care has progressed and continues to do so. The information shared on CAR-T therapy and the new and innovative ways in which we're able to monitor and gather data from patients is really exciting. Thank you so much for your insights. Lastly, I'm very excited to introduce Dr. John Mascarenhas as our speaker covering innovation in myeloproliferative neoplasms. Dr. Mascarenhas is a hematologist-oncologist at the Icahn School of Medicine at Mount Sinai and a member of the Tisch Cancer Institute. Dr. John Mascarenhas is one of the leading figures in the MPN space. He's published over 275 papers during his career, been invited to speak for countless different conferences and events around the world, and has served as a principal investigator of numerous clinical trials centered around innovating treatment approaches for MPNs and secondary leukemia. Of particular note, Dr. Mascarenhas serves as the principal investigator for the clinical trials arm of the National Cancer Institute-sponsored Myeloproliferative Neoplasm Research Consortium.
Hi, I'm John Mascarenhas from the Icahn School of Medicine at Mount Sinai. Today I'm going to be reviewing what's on the horizon for myelofibrosis. But before we talk about what we're at the leading edge of, I want to just review for everyone where we've come from. So in 2005, with the discovery of the JAK2 V617F mutation that was found to be one of the main pathologic drivers of the JAK-STAT signaling system that contributes to the pathology of this disease, it really was the first molecular insight into this disease process that then led to the advent of JAK inhibitors, which has really transformed our field in a significant way. So the first JAK inhibitor that was approved was ruxolitinib in 2011, then followed by fedratinib in 2019, pacritinib in 2022, and then ultimately momelotinib in 2023. So in 2024, moving forward, we now have in the US 4 JAK inhibitors at our disposal. Now, they all have in common that they're JAK2 inhibitors, but there are differences in the kinome profile that likely explain differences in both their activity and toxicity profiles that allows for a practitioner to really tailor the therapy and personalize the approach with sequential options as well that didn't exist 10 to 15 years ago. Ruxolitinib still remains the most useful and used upfront JAK inhibitor for the majority of our patients. But for patients with very low platelets, less than 50,000 or even less than 100,000, we have pacritinib. And then for patients with very significant anemia and particularly transfusion-dependent anemia, even upfront, we can even consider momelotinib. So with these four options, one must understand that although they are all JAK2 inhibitors, due to sparing of JAK1, for example, with pacritinib, and to some extent with fedratinib, that may change some of the efficacy readouts that we see, less myelosuppression with pacritinib. With the fact that both pacritinib and momelotinib are ACVR1 inhibitors, we've learned that that likely mediates hepcidin expression and improvements in hemoglobin that we see with pacritinib and momelotinib, but less so with ruxolitinib and fedratinib. And then the fact that both pacritinib and fedratinib are FLT3 inhibitors likely explains why the toxicity profile with more GI-related nausea and potentially diarrhea are seen with those agents compared to ruxolitinib and momelotinib. The only drug that has a black box warning is fedratinib with Wernicke's encephalopathy, so please check vitamin B1 thiamine levels and replete as needed. But all of these drugs can really be tailored, and there's overlap. They're not, mutually exclusive in the upfront and sequential setting. But where we're really moving forward to, and joining the rest of oncology, I would argue, is combination therapies. For the rest of the time, I'm just going to highlight some of the exciting advances in combining rational therapeutics. And what's nice about everything I'm showing you is it's all predicated and endorsed by preclinical modeling that supports the clinical evaluation of these compounds. So the first one I want to highlight today is the pelabresib MANIFEST-2 phase 3 study, which is an evaluation of an oral pan-BET inhibitor, pelabresib. And this was in combination with ruxolitinib versus placebo in myelofibrosis patients who were intermediate 1 or higher risk and were JAK inhibitor naive. And the primary endpoint was spleen volume response at 24 weeks and total symptom score at 24 weeks. And the concept here is, can we do better than single agent JAK inhibitor? Can we get deeper responses from a spleen and symptom perspective? And then does that translate to better durability of response? And then ultimately progression-free survival and maybe even overall survival, which obviously requires much greater follow-up with these studies. And I'm highlighting this study because I think it really is it's a potential for a paradigm shift, but also highlights some of the challenges in developing therapeutics in myelofibrosis. So you can see from this waterfall plot that there was a clear advantage of the combination of pelabresib plus ruxolitinib in almost doubling the spleen volume response, 66% versus 35%. And that dark blue shows you that difference there. What was not statistically significant was the symptom improvement, which I'm showing you here. So although the p-value is very close, 0.0545, and the delta between the two was a mean difference of 2 points on the scale, it was not, as proposed in the trial, was not statistically significant to be a winning endpoint. So it necessitates the longer term follow-up of these patients to see what happens at latter time points, 48 weeks and on, and do patients stay on these drugs because their responses are durable and more meaningful. It also really highlights the challenge of improving upon symptoms beyond what ruxolitinib does alone. Ruxolitinib as a JAK-STAT inhibitor is very potent in downregulating inflammatory cytokine signaling, and this, we think, underlies the significant symptom improvement we see in patients. And that's probably one of the main reasons why patients live longer too. They feel better, they move, they can eat, they reverse cachexia, and they are likely to have an improved survival, even simply from symptom improvement. And if you have a ceiling effect, which we see particularly with fatigue, it's really difficult to do better than that. If one looks under the surface, and again, I'm using this study to highlight what we're looking at in terms of biomarkers, we're seeing with these agents, and here with pelabresib, a greater degree of reduction in bone marrow fibrosis at 24 weeks, and less worsening of fibrosis over time, suggesting disease modification. Pelabresib hits NF-kappa B, which is a main regulator of a lot of the inflammatory cytokines, has been shown to be upregulated in myelofibrosis. And this is done more potently with the combination of pelabresib and ruxolitinib than ruxolitinib alone. So there's very significant modulation of biomarkers associated with the disease. It is really paramount as investigators, sponsors with the FDA to validate that these biomarkers are actually clinically meaningful. And that's sort of the unmet missing piece here, is that we have a lot of this data, and this is one example. We saw data with navitoclax, the BCL2 XL inhibitor, as well as many other agents that they can have effects in histopathologic features, molecular features, and cytokines. But we need to validate that these actually correlate with outcomes that actually matter, progression-free survival and overall survival. To that point, I want to point out an interesting analysis done with selinexor, the XPO1 inhibitor, approved in myeloma and other lymphoid diseases, but here we're using it in myelofibrosis based on preclinical data that would suggest that it hits many relevant pathways, including p53. And this drug is given in this early phase study once a week at either 40 or 60 milligrams daily. And we saw very significant spleen and symptom responses. But what I'm showing you here is the correlation between reduction in certain cytokines that are known to be pathogenic and drive the disease, like TNF alpha, IL-18, IL-7, and the correlation with a clinical measurable change in spleen volume. So correlating reduction in cytokines by on-target activity with a clinical outcome measure like spleen volume. So this has been an emphasis in many of the trials. Now this drug will move forward in a phase 3 study in the JAK inhibitor naive population of selinexor at 60 milligrams once weekly, plus ruxolitinib versus ruxolitinib plus placebo in patients who are JAK inhibitor naive. On the same theme, I want to show you data from the navtemadlin studies. Navtemadlin is a very potent first-in-class MDM2 oral inhibitor that upregulates p53 pathway in wild-type p53 patients. And here we're showing in an add-on strategy to ruxolitinib patients who have a suboptimal response, not just the improvement in spleen and symptom, but here I'm specifically showing you another example of biomarker modification, significant reduction of circulating CD34 cells, as you can see, that deepens with time and then correlates on the right with, again, an outcome measure that we can measure by imaging, spleen volume response. So the attempt to correlate these changes in biomarkers with clinical outcome measures that don't take necessarily many months or years to read out. And I think this is really critical that we demonstrate this. I show this slide to show you where where we're moving as a field beyond the upfront setting. This is an important study called the POESIS study, which is taking JAK inhibitor naive patients, providing ruxolitinib for a certain period of time, and then assessing whether they are responders, suboptimal responders, or non-responders. So this is a study where it's just taking the suboptimal responders, those patients that still have unmet need from a spleen and symptom perspective, and then randomizing them to the addition of navtemadlin versus placebo. I love this study design. I think this is a very clean, very strong effort to demonstrate the added benefit of an agent like navtemadlin to a group of patients who haven't gained sufficient response by criteria that are used for regulatory purposes to compensate. So this is a study now that's opening globally and enrolling, and will be an important study to keep one's eye on. On the same theme again of disease modification and biomarkers, this is data from the Metelstat phase 2 randomized study, 2 different dose levels in patients who are relapsed refractory after JAK inhibitor therapy. This is a patient population we know unfortunately has a poor outcome, that we're interested in improving survival. And here we saw from this study that at 9.4 milligrams per kilogram, the higher dose, we had a survival that was close to 30 months versus almost 20 months in the 4.7 milligram per kilogram, which looks better than the 12 to 14 months historically in 5 different studies. So for the first time, we're seeing an intervention with a drug, an intravenous drug, not oral, given every 3 to 4 weeks, that does seem to improve survival in patients. Here we're showing you on the left a correlation between VAF reduction of driver mutation and survival in red of at least just 20%, so not even 4.5 log reductions, but even a 20% reduction in VAF and improvement in survival. And then on the right, bone marrow fibrosis reduction and survival. So correlating biomarkers with here survival rather than spleen volume response. And this is a study now that has informed the phase 3 study, which I'm showing you here, the IMPACT-MF study, taking patients who are refractory relapsed after JAK inhibitor randomized into imetelstat every 3 weeks versus best available therapy excluding a JAK inhibitor with a primary endpoint of overall survival. The only study that I'm aware of in MF in which an ambitious endpoint like survival, which is obviously important, is being utilized. So the field is truly moving. You can see it's moving in different ways, and it's moving from combination therapies up front to salvage therapies to treatments that might improve overall survival. And there's a number of other drugs I haven't mentioned, like TP3654, the PIM1 kinase inhibitor that looks promising, that are all now moving into later stages of clinical studies. So I definitely would emphasize and reiterate that it is essential that our patients with myelofibrosis are preferably treated on studies so that we can better understand how to move these drugs forward and integrate them to improve our standard of care. Thanks very much.
Thank you, Dr. Mascarenhas, for sharing this presentation with us. It's incredible to see how far treatment for myelofibrosis has come in the last 20 years, and it sounds like using them in combination with other therapies is an exciting new approach.
We're so grateful for these amazing doctors and their sharing of this. And now what we'd like to do is I'd like to share a little bit with you about our innovation, HealthTree's innovation, and show you HealthTree Care Hub. So let me do that. As we learned from these doctors, your participation and our participation as patients in clinical research, joining clinical trials, is really essential. But not all patients can join clinical trials. So in some childhood cancers, clinical trial participation is in the 80% range because parents are usually picking their children up and going to a child-based cancer center. And for adult cancers, that's a little bit trickier when you have families and homes and things like that. So there are only about 5% of adult patients who participate in the clinical trial research. And as you heard from these doctors, this is how they bring new drugs to market or to us in the clinic. And they help us find new and better treatment options that are going to give us better outcomes. Something else that can help is something that is called real-world data. And this is a whole new category of research that has not been done significantly in the past just because it was so challenging to do. So this is providing data for research. And as we have more options, and especially as we see hundreds of options or 20 different types of drugs, the need to aggregate data quickly is going to be better than running a series of 10 clinical trials to compare different drugs together and things like that. So what does innovation mean to us? Innovation means, number one, improving patient outcomes. So our patients living longer and using these treatments in better ways. Innovation to us means personalizing care. So as you heard, several of these doctors talk about specific personalized care, either from genetics, or that could be based on age, or that could be based on just different subgroups. The goal is to personalize care in these subset groups. That means implementing new models, tools, and technology. And healthcare traditionally has been lagging significantly in its ability to apply technology into research capacities. So what HealthTree is doing now is changing that. We want to speed the pace of research. To me, I'm a patient. I feel like I don't have time to wait. The clock is ticking, and there's a sense of great urgency around what we are doing for the patients and for the doctors, because we won't all last, a very long time and decades with some of these diseases. So how can we as patients help them cure us faster? And that is where our focus is. So today's challenges, doctors have really limited access to data today. A doctor at a center like you heard, Mayo Clinic or Mount Sinai, has access to their patient data. If their patient is, for example, getting labs run at Mount Sinai and is getting other labs run down the street, the Mount Sinai doctor does not have an updated view of all that patient data. So that's the challenge. Facilities can't really share their data because of HIPAA protections, and research today is very, very slow. We're digging into the need. We've dug into the needs of patients for the last 12 years and have tried to satisfy patient needs for education and community building and connection and all of that. And now we're working to solve patient problems of cures by helping the research community get what they need and in the ways they need it. And today, if someone wants to do a research project, they will manually extract data out of their electronic health record system from their patients. They'll manually enter it into a database. Then they'll transfer it to like a CSV or Excel file and try to run statistics on it. We want to shorten up that process considerably and give them access to not only their patients' data, but anonymous data from more patients. So patients, we can turn that model upside down. So I'd like to share a few concepts about this HealthTree Cure Hub 360-degree registry idea. So patients, we can share our information with researchers. We can provide more complete data than they have access to today. We can connect all of the records from all of the places we are going to see. So I've been a patient at 4 different centers. I can aggregate all my information into a single patient profile, and then I can open that back up to my doctors for research. The completeness of the data also includes our side effects. Sometimes when we go into the clinic, we'll report maybe the top 2 side effects that we're having and the severity, but sometimes it's not recorded properly in our chart. And it's definitely not all of the side effects are because, we have 15 minutes on our appointments. So that you just don't want to burn up your entire, visit time talking about all of your side effects when you're also talking about, okay, what's my next step? So we have the ability to aggregate that data too. Now, researchers can be provided access to anonymous patient data. And this data is secure and only used for research purposes. So a lot of times people will ask us, who are you? Why are you doing this? And what are you going to do with my data? I would say those are the most 3 commonly asked questions about HealthTree Cure Hub. And we are patients just like you. We are trying to make this research process significantly faster and provide better tech tools for patients and researchers. And we are trying to achieve cures. That's our goal. And this includes a network of over 2,900 hospitals. So with HealthTree Cure Hub 360-degree registry, you don't just participate in your care, you are really changing the face of cancer research. Here's an example of what we've been able to do so far in the multiple myeloma space. We've done over 76 studies involving over 65,000 participants and have involved over 78 investigators. Our recruitment time, because we have connections to patients through multiple ways, through our education programs or our community building programs, we invite you to participate. Many of you have been incredibly gracious to do that. You are helping these researchers move data forward faster. And we're able to ask questions that they would never have asked before because it was just too laborious or too expensive. To give you an example, I asked one of the doctors that we did a survey for how long this 10-question survey would have taken. And we had 1,500 of you respond to this survey. And he said, well, I would have not had 1,500 responses from my center. So I would have had to do a cross-sharing data agreement with another center. That would have probably taken me 6 to 9 months to obtain that. I would have had to hire a research coordinator. And he said overall, it would have saved him 1.5 years and $150,000. And we did that research survey for him in 4 weeks, and we gave him the data for free. And it was all just aggregated, anonymized data, and he was able to go and continue on his path for research. So that's what we're trying to accomplish with this product. Now, what's the power of disruptive innovation? We are breaking through with slow traditional models. It's shocking that in 2024, the doctors are using, and the researcher community are using these very slow, very manually oriented methods. We want to aggregate complete data and open it up for them. We have new tech tools. So like I mentioned at the beginning of the program, we have built 14 different software tools to enable all of our advocacy work and this data portal. And our disruptive innovation makes sure that patients are the driving force behind research and treatment innovation, and that we're not just passive recipients. I know that when I got diagnosed, the average age of myeloma diagnosis is about 72. I was 43, and I felt like I don't want to just last another 10 years. I need another 20 or 30 years because I'm just not satisfied. So I've dedicated really the rest of my life to doing this and to making this go faster, this research process go faster. So by participating in HealthTree Cure Hub 360-degree registry, you're advancing research not just for yourself, but for every patient that comes after you. So with that, I think I will show you a demo of how this actually works. So this is HealthTree Cure Hub. And in HealthTree Cure Hub, you can connect your medical records to one or multiple facilities. And this is my patient profile. You can see that I have, I'm seen at multiple cancer centers. If I want to create a new connection, I just type in, let's say I'll recreate my Huntsman connection here. This is where I go for most of my chart or most of my appointments. It'll bring me to my MyChart login, and depending on the type of system you use, so we have integrations with MyChart, with Epic, with Cerner, with the VA systems, and we're working on our Meditech integration, which is about 95% of all hospitals. And we have integrations right now with over 2,900 hospitals. So I sign in. HealthTree does not save your login credentials. You're just signing into your regular MyChart. We say what we will and won't do with your data. You can select or unselect these things that you want to share, and you can say for how long you want to share them. And we request that you do the longest period of time. And then it will go and pull information from the starting point when you were at that facility. So this will pull 14 years worth of my data into a single system. Then we have a team on the back end that can go in and validate things, because sometimes things like your FISH test or your genetics test isn't coming across as structured data, it's coming across in a PDF, and we want to add that to your profile. So then I can add another facility and I can close it down. But let me just show you what type of information that's pulling. So it's pulling over from my chart, my diagnosis. It's pulling over from my chart, my labs. And this is 14 years worth of my labs. You can see how I'm tracking all my labs. We specify when we do this, we started this in multiple myeloma, and when we have created this for other diseases like CLL, MDS, AML, and myelofibrosis so far, we are mapping the disease-specific labs that you need to track in this system. So you can see it like this, or you can see it in chart mode. And this is a nice way for people who like keeping spreadsheets of their labs. And you can see it's tracking all my key myeloma markers. And it's taking a minute to load, but you get the idea. It's also tracking my genetics. So when our medical team reviews a chart, they will pull things out of the profile to be able to put them in there. So we know what type of genetics that they're working with. I know in AML, myeloma, this is very important. AML is extremely important. So you can kind of see it will show percentages. We'll take the notes and include that there. Also your prior treatments. And you can see it's organizing it by my line of therapy. That's what they call it. So my first treatment was, I had this tendon stem cell transplants, and you can see like the dates of when I had all that. And then my most recent was a CAR T therapy. So you can see how I received that and the things that were part of that. And then it goes on to do my imaging, my care team, and a disease summary that you could print out and take to another location if you wanted to. Now I'm going to jump to this accelerate research because this is a way that you can help accelerate cures. In here, we partner with myeloma investigators, and I'll use this as a good example. Bispecific antibodies, you heard this from multiple of the doctors that just presented across all blood cancers. The bispecific antibodies are this very new and interesting area of research. So we have 3 approved bispecific antibodies in multiple myeloma, and everyone wants to know, like, what's the best sequence? Should we do CAR T before or after? What about the side effects? Who are getting side effects? Who are not getting side effects? Who are responding and who are not responding to these diseases? What did they have before? What are they doing later? So we have partnered with, you can see an extensive number of investigators and the International Myeloma Society as an organization. And we're asking you, if you have had a bispecific antibody, please join this study. We'll be recruiting for 3 years. We'll be pulling the data, and it's a way to have hundreds of patients join this. This is research that would not be done inside of a clinical trial because the clinical trials are really designed to be able to get the drug approved. And then once it's approved, there's not a lot of research that happens about, well, what else happened, after it became. And for the people inside the clinical trial, is it the same as people who were not able to join the clinical trial? Did they have more or worse side effects? Did they live as long as the clinical trial data? So this is almost like a phase 4 study for this. We have several studies in here that are cross cancer. So a physical activity study with a Memorial Sloan Kettering Cancer Institute or Cancer Center investigator. And you can see that we have other cross-cancer surveys as well. A UT Southwestern doctor wants to understand sexual health of blood cancer patients. We have anything from understanding the stem cell collection process to your stem cell transplant experience, use of supplements. We will be launching a CAR T study shortly to do the same thing as in bispecific antibodies. And that is how so an investigator will come to us, have an idea for a survey or a study, and then we implement it, we put it in here, we invite patients to join, and then we turn the data around and give it to them so they can further the research. We also have other features in HealthTree Cure Hub where you can see treatment options. We're working on an AI-based clinical trial product right now. So because we have information about labs or your creatinine level or your number of lines of therapy that you've already had, our goal is to develop a personalized clinical trial finder so you can see clinical trials that you personally are eligible to join. And then we have a side effect solution finder. So if you struggle with a side effect, let's say, for example, neuropathy is pretty common in myeloma, you would be able to report and see what other people are experiencing and what they're doing to solve these problems. And this is available for all of the diseases that we are working with. So you can see that we just want data behind this. And it's better than just a shout out on Facebook and then hoping you get some good information. The other thing that patients can do in this, and this is what I wanted when I was diagnosed, they can find their twin. It's going to take a minute for me to load, but the concept is, and what I wanted to see when I was diagnosed is show me all younger patients, show me patients that have the same genetic features that I do, show me what they got for therapy, and then show me how they responded, because wouldn't that be nice to be able to see several years ahead to the best potential outcome? And that data was just not available to me. So I can find patients that are very similar matches to me, and it's all de-identified, so you can't see who that person is. You can see that it was pulling based on some genetic features. I could add some criteria. I can see what this person did for therapy, and then I can send them a message. So with that, let's go back to the end of our presentation. And just thank you for participating. So we invite you to join HealthTree Cure Hub and this new idea of this 360-degree registry. We will be partnering with individual institutions to be able to turn data around for their doctors, because they do not have access to this level of data, and we want to provide solutions to you as patients and to them, because it will all mean cures for all of us together. So these are our upcoming blood cancer events. We thank you so much for participating. Again, a special shout out to GSK for supporting us during Blood Cancer Awareness Month. And with that, we just want to thank you for joining and participating.